Today, we're going to talk about a piece of technology that is so fundamental to modern medicine, it's practically invisible. It's this, a simple petri dish filled with a kind of pinkish gel called MacConkey Agar. It might not look like much, but over a century ago, this thing completely changed the game in public health. So how did it do it? Okay, I want you to imagine for a second that you're a scientist back in the late 1800s. Your city is getting hammered by diseases like typhoid and cholera. You know they're caused by tiny germs, probably in the water supply. But the problem is, when you try to grow a sample of that water, you're faced with this just a chaotic soup of millions and millions of different bacteria. How on earth do you find the one single culprit that's causing all the chaos?
And that right there, that needle in a haystack problem, was the huge crisis facing public health. I mean, cities were exploding in size, sanitation just couldn't keep up, and even though scientists knew germs were to blame for these deadly outbreaks, their tools for actually finding those germs, well, they just weren't working.
So here was the real issue they were up against. When they'd put a water sample or a sample from a sick patient onto a culture plate, the normal, everyday bacteria, the harmless ones, grew so fast and so thick that they would just completely overwhelm the entire plate. And the dangerous pathogens, the actual needle in the haystack, they were totally buried, totally hidden. This made diagnosing an illness or preventing its spread almost impossible. So, this is where our hero, Alfred MacConkey, steps onto the scene. He was a Scottish bacteriologist, and his job working for the Royal Commission on Sewage Disposal was to solve this exact problem. He desperately needed a better way to hunt for the specific germs that came from the gut.
And his big idea, well, it was one of those brilliantly simple things that makes you wonder why no one thought of it before. MacConkey just reasoned that any bacteria that actually lives inside our intestines has to be tough. It has to be able to survive the really harsh, bile-filled environment of our digestive system. Most other bacteria, they just can't handle it. And that single, simple insight, it changed absolutely everything.
Now, it wasn't an overnight success story. It was a process, of tweaking and refining. He started in 1900 with just a liquid broth that had those key bile salts in it. A couple of years later, some colleagues added crystal violet dye, which knocked out even more of the unwanted germs. And then finally, by 1905, he nailed it. He perfected the solid agar plate that we still use today, the one that lets individual bacteria grow into these nice, neat, separate colonies that you can actually see. So what makes MacConkey Agar so brilliant? It's that it isn't just a plate of food for bacteria. It's more like a cleverly designed obstacle course. It has two incredibly important functions that work together perfectly: selection and differentiation. All right, first up is selection. And the easiest way to think about this part is to imagine the agar is a bouncer at a super exclusive nightclub. He's got a very strict guest list, right? And his entire job is to keep the wrong crowd out. And here's how that bouncer actually works. See, bacteria are basically split into two big groups based on their cell walls: gram-positive and gram-negative. The bile salts and the crystal violet dye in the agar are toxic to gram-positive bacteria. They just destroy their exposed cell walls. But, and this is the key part, the gram-negative bacteria that you find in the gut, they have this extra protective outer membrane. It acts like a shield, blocking the bile and the dye, and so only they are allowed to grow. Pretty clever, right? Okay, so you've made it past the bouncer. But that's not the end of it. Now comes part two, differentiation. Once the right kind of bacteria are on the plate and growing, the agar puts them through a little test to reveal who they really are. And this test is all about what's on the menu. The main dish being served is a sugar called lactose. Now, a lot of harmless gut bacteria, like most E. coli, they love lactose. They ferment it, which releases acid. This acid then triggers a special pH indicator that's mixed into the agar, and bam, their colonies turn this bright, vibrant pink. But the really dangerous pathogens, like salmonella, they can't eat lactose, so they eat protein instead, and their colonies stay colorless. And just like that, the bad guys stick out like a sore thumb. You can't miss them. So let's look at the full recipe, the whole crew working together. You've got your basic nutrients, so things can grow. You've got the lactose, which is the test fuel. You've got not one, but two bouncers, the bile salts and the crystal violet, for security. Then there's the neutral red, that's our color referee that calls the plays. And finally, the agar itself, which is just the gel that holds the whole thing together. Every single ingredient has a very specific and very elegant job to do. And what's really cool is that the original recipe was so genius, scientists realized they could tweak it for special missions. By just swapping out an ingredient or two, you could create a brand new version of MacConkey Agar to hunt for a very specific culprit. So take MacConkey Agar, for example. In this version, the lactose is replaced with a different sugar called sorbitol. And why would you do that? Well, because while most E. coli are harmless and they have no problem eating sorbitol, that really nasty O157 strain, the one you hear about in bad food poisoning outbreaks, it can't. So on this special plate, the color code is completely flipped. The harmless E. coli turned pink, and the dangerous O157 stands out as a suspicious colorless colony. Or check this trick out. There's a type of bacteria called Proteus, and it's a real pain in the lab because it swarms. It just spreads across the entire plate like a big, messy blanket, hiding everything else underneath. But scientists discover that if you simply remove the salt from the MacConkey recipe, Proteus can't swarm anymore. It's forced to grow in nice, neat little colonies, revealing any other bad guys it might have been hiding. So, okay, this is a great story from over 100 years ago. But why should you care about it today? Well, the answer is everything. This century-old invention is still a critical frontline tool, quietly protecting our health every single day. If you've ever had a UTI, I can almost guarantee the lab used MacConkey Agar to figure out what was causing it. It's the first test labs run to screen for salmonella in food poisoning cases. Our drinking water and food supplies are tested with it to check for contamination. Even the pharmaceutical industry uses it to make sure medicines are safe. Seriously, this stuff is everywhere, working behind the scenes. You've probably heard the term coliforms on water safety reports. Well, this is what they are. They are those gram-negative, pink-turning, lactose-eating gut bacteria that MacConkey Agar is perfectly designed to find. Now, they're not always dangerous themselves, but finding them is a huge red flag. It means that the water or food has likely been contaminated with fecal matter, which means much more dangerous germs could be hiding in there too. When you boil it all down, it's that simple, elegant logic of MacConkey's plate. The bouncer, to keep the wrong germs out, and the color-coded test to identify the ones that get in, that has truly stood the test of time. It's cheap, it's reliable, and it gives scientists a clear, visual answer, usually in less than a day. It's just a perfect example of the incredible power of a simple, brilliant idea. And it really makes you wonder, right? What other simple, 100-year-old technologies are still out there, working behind the scenes, silently keeping our modern world safe every single day? Thanks for sticking with me on this one.